Next phylum, phylum Amoebozoa. Is that right? Most of the amoeba are in this group. What else? Amoeba. There are a few species I listed for you. Entamoeba histolytica that I said it causes amoebic dysentery. Usually, if little children in third world countries they get it, there is no medical attention to them quickly, they might die. But here in the United States, of course, the difference between dysentery and diarrhea. Diarrhea contains no blood, but dysentery is the diarrhea with blood. Okay, I hope you all know what I'm talking about. So it's diarrhea with the blood is called dysentery. So Entamoeba histolytica cause amoebic, they call it amoebic dysentery. And then Entamoeba coli, now I thought I fixed it. No? Yes, it's a commensalism. Yes, it is a commensalism. How come you guys didn't say that in your discussions? And hard to distinguish between Entamoeba coli and Entamoeba histolytica. But some people, some scientists say, you know, after, of course with DNA you can distinguish between the two, but under microscope it is difficult. So, and then the other one, Entamoeba gingivalis, your textbook referred to it as opportunistic parasites. 50% of population have it. How is it transmitted from one person to the next person? Is by kissing. Okay, that's another reason I don't like kissing, right? When you kiss somebody, you're kissing a tube half full of what? Poop? Is that right? You're kissing something or somebody, a tube half full of poop. Rem, you probably don't want to kiss anybody anymore, right? Okay. So anyhow, Entamoeba gingivalis, opportunistic protozoa. Again, as I said earlier today, what it caused if a person does not keep a good hygiene, do not brush teeth, do not take care of it, then these organisms can cause gingivitis. And gingivitis is inflammation of their gums. Right? Am I making some sense a little bit? The other one that are kind of amoeba, but before they were in this phylum, but now they separate them and put them in their own phylum, is phylum Foraminifera, which is shelled amoeba made up of calcium carbonate. They have these tests. It's made up of silica or silt. So test, T-E-S-T, is the shell of these organisms. So inside, just like a snail, inside of the shell there is an amoeba, single cell amoeba. You know, snail is multicellular organism, but amoeba inside of these tests, the shell, exoskeleton, whatever you want to call them, but usually referred to them as a test. Inside of the shell is a one-cell, single-cell organism called amoeba. And then the other one, phylum Radiolarians and Helizoans. They are made up of silicon, same thing. And we do have slide of them in the lab of Radiolarians. We do not have any Helizoans, but Foraminifera and Helizoans and Radiolarians. We have them in the lab, and you should be able to distinguish them in the lab practical exam. You should be able to distinguish them which one is which. Okay, so which one, and they are all different shapes. You know, these shells, these tests, you're looking at them on a microscope, they all have different shapes. Okay, and then phylum Dinoflagellata, I'm mentioning it last, it's a serratium. That's why I put it in here. Serratium is the one we do have in the lab. And the next one, which I'm going to talk about, I guess red tide. Well, here is the amoeba. Here are the Radiolarians, Foraminophorans, Helizoans, right here. So they all have different shapes, and the amoeba lives inside of the shell. This is the amoeba that we have in the lab, and it does not have a shell. So don't think all amoeba have a shell. We have the amoeba in the lab that does not have a shell. The other one, Acanthamoeba, that none of you mentioned it this morning. Did I see? Yes, is that right? I thought we have some slides in the lab, but I guess we do not have the slides. Contact lenses, usually the culprit is contact lenses. People who have contact lenses is transmitted to their eyes, but of course they do not keep a good hygiene of their contact lenses, and that's why they get it. Okay, you're supposed to wash them, you're supposed to take care of them. You all know, all of you that have contact lenses, you know what I'm talking about. You're supposed to take care of them. But if they don't keep a good hygiene of contact lenses, then it is possible that they get acanthamoeba. Showers, contaminated water, not human to human. Usually it's not, if you rub your eyes against somebody's eyes, raw eyes, usually that does not happen. So it's human to human, does not happen with acanthamoeba. And then FLA is my abbreviation for living amoeba infection like Naegleria fowleri, acanthamoeba, castellina. These are the two organisms that are FLA. It means they are free living. However, when they come in contact with human, they can cause problem. Okay, and then I will talk about Naegleria fowleri here in a minute. So these are, again, acanthamoeba is also in phylum, not this one, not Naegleria fowleri. Naegleria fowleri is not in phylum Amoebozoa. Okay, you will see it. You already saw it. You studied your notes. But acanthamoeba is in phylum Amoebozoa. Okay, phylum Heterolobicea, Naegleria fowleri is a naked amoeba with eruptive pseudopod that forms abruptly and sometimes called amoeba flagellates because you will see, I show you the life cycle. Remember, at a certain stage of the life cycle, this organism develops flagella. Okay, it's a life cycle. At some time, they have flagella. So schizopyrenoid, since life cycle includes both amoeba and flagellated stages. Primary, the disease that it caused, you mentioned it this morning, primary amoebic meningoencephalitis. It means inflammation, inflammation of your meningitis, meninges, I'm sorry. Meningitis, it means inflammation of the meninges. Meninges is a layer of epithelial tissue covering up your brain, your spinal cord. And this is an organism, can cause inflammation of it, but other organisms, bacteria, viruses, can cause meningitis. But this one is called PAM, abbreviation for it, PAM, primary amoebic meningoencephalitis. Flagellate and amoeba trophozoites, binary fission, of course, transmission by amoeboid form, and death within 5 to 7 days. If somebody gets it within 5 to 7 days, that person will die. As simple as that. Okay? So diagnosis can be difficult. You cannot send the patient home. If you become a doctor, you cannot send the patient home and say, go take some aspirins, you'll be all right. Okay? So you got to ask him a question. Have you been swimming in semi-dirty water? It has been found in swimming pools too, that they use chlorine. So you got to ask them these type of questions. And if they have been swimming, and then maybe you should, not necessarily in a pond, a lake, or dirty water. As I said in baptism, when they submerge in water, kitchen sinks have been found. So they've been found in places, but you got to ask the question and then keep up with that patient. If after a day with aspirin or 2 does not go away, then maybe immediately on day 3 or 4, immediately put them on these antibiotics. Intravenous, these are the drugs that are used. And here's the life cycle, the amoeba. Again, do not worry about these drugs. I never ask you the name of these drugs. I just thought I'd put it here. Someday, you say, Amir talked about this. You refer to your lecture notes, and then you have the drug in there. So that should help you. But no, anyhow. Amoeba, transmission transformed to the flagellated organism. It can form a cyst. So, and then we human ingest it while we're swimming through our nose or through our mouth. They end up in the brain. Okay. Here is a picture that they're destroying the cells. They poke a hole inside of the cells. Is the amoeba, and then of course there are signs around beaches sometimes that say it's amoebic meningitis, and then they warn you that you should stay out. This is the one you have in your 2x2s. This is the slide you have in your 2x2s for this exam. You do have 2x2s slides also. So this is the one that is for nuclear-farine brain impression. How to get nuclear-farine ponds and lakes, water into nasal passage, immersion in bath water, playing in warm, muddy puddles after rain, during summertime, placing head in a trough of water on a school playground. These are all cases have been reported that transmit your nuclear-farine. In some baptism, I already talked about that, the full immersion also called out. Okay, the next phylum. Phylum Viridoplantae, you are dealing with the organism right now. Your experiment is phylum Viridoplantae. So Viridoplantae they're unicellular, colonial. The one you're dealing with is colonial. Your volvox is colonial organism. And multicellular green algae that used to be the phylum Chlorophyta. This group includes vascular and non-vascular plants too. So you know, your textbook says something nice about the classification. When we talk about the classification, it depends who you are referring to. Okay, so who you're talking to, what classification they believe in. It's so diverse now, the classification, but we've got to stick to something. That, you know, part of this class is to learn the classification. So that's why I bring it up, and I'm hoping you guys would know this. Clade Plantae, so this belongs to Clade Plantae. The Viridoplantae is the name of the phylum, belongs to that phylum. Chlamydomonas, when I was in college, when I went to Kentucky, we had them in the lab. I should ask for them maybe next time. They're a little neat organism. They're kind of huge. You can see them on the microscope nicely. So it has chloroplast, it does photosynthesis, and it's a nice organism. It's flagellated, okay, but maybe next semester. Volvox, you're dealing with it right now. It's a colonial. It's a colony of cell right here. I mentioned that. Here is a life cycle of Volvox. Have cell wall, division of labors. It means some cells are responsible. What I mean by division of labors, some cells are responsible for photosynthesis, making food, and some cells are responsible for reproduction, sperm and egg. Okay, so the cells that are responsible for photosynthesis, making food, are called vegetative cells. I'd like to, and then in your slides, make sure I ask you on slide description, make sure you identify the vegetative cells. So the cells right here are, all of these cells are vegetative cells. And this is a flagellated organism. A lot of you make a mistake during exam. This is a ciliated organism. No, it's not ciliated, it is a flagellated organism. They have, each individual of these cells have flagella. Life cycle of it again, sexual, this cycle is a sexual life cycle. This is asexual life cycle, and what I mean by asexual life cycle, inside of each colony, there are new daughter colonies are formed, and these new daughter colonies become bigger and bigger and bigger, and they leave mother colony. They leave mother colony, and either the sexual reproduction can happen in them, or they can become inside of them, another colony grows. What else? Asexual sexual reproduction, of course, I talked about that. Macrogametes loaded with food, they are called macrogametes, and microgametes form sperms to find the ovum. And zygotes multiply and break away, just like they're showing it to you here, and then the zygote can become a colony. Okay. Phylum Diplomonada, a mitochondrial lacking. They do not have mitochondria. The members of this phylum, okay, lack mitochondria but have mitochondrial genes in the cell nucleus. Okay, as you know from Bio 1, that you know mitochondria have their own DNA. Okay, you know that from chloroplast, mitochondria, nucleus. These three organelles you learn from Bio 1, and one thing, another organelle you learned in here, kinetoplast. They have their own DNA material. Okay? So, what this sentence is saying, they do not have mitochondria, but they have the DNA, the gene, in the nucleus that are similar to the mitochondrial DNA. Okay. Giardia, there are two species. I would like to know in the lab, you cannot identify them, but there are two different species. Some scientists, I don't know, I do not keep up with the reading anymore with these guys, so believe that they both are same. Okay, some scientists say no, they're two different species, but some scientists say no, they're the same. But anyhow, you should be familiar kind of with both names, Giardia intestinalis and Giardia lamblia. Here's the life cycle of it, if you would. This is what you have on the microscope. You have this, of course, under your microscope, under 40x, they're very small. Some of you need some assistance. I don't mind to help. But anyhow, and I would ask that during lab practical exam, historically, if you can ask those guys, have you asked for Giardia? No, they're so small, they're hard to find. Yeah, they show up in the lab practical exam. Okay, so anyhow, let's go over the life cycle. Giardia, lamblia, flagellate, trophozoite. Trophozoite in this class, you guys, means feeding stage. Cyst is not a feeding stage, you know that. A cyst, all cysts, they are not feeding. So, what happens with trophozoite in large intestine of us or other animals, okay, cow, sheep, pigs, so multiply asexually by binary fission. You all know what binary fission, I talked about that. Okay, and then what happens, diagnostic infective stages, resistant, so you see that in feces, the cysts passed in feces, trophozoites may be found more commonly in slots of the fluid. And then methods of infection, man ingests infective cysts transmitted by feces, finger foods, and fomites. Do you guys see that term? I'm hoping you would know what fomite is. A fomite, this is a fomite. Your keys are a fomite. Your pen or pencils, inanimate objects. That's what the definition of fomite is. Inanimate objects that can transmit a disease. A pathogen, if you would. That was in your quiz, or afternoon lab quiz. Pathogen, what is the definition of pathogen? Giardia is a pathogen. Right? Am I making some sense? Vanessa? Yes? Okay. So cyst pass to small intestine, excistation, you know excisment occurs. So what happens, and then of course it goes to the intestine of the host. So what happens when a person becomes infected? Let's start with large intestine. In the large intestine, small intestine, these guys are multiplying, right? And then it's time for them, then I have diarrhea, right? They cause diarrhea. Then I have diarrhea. In my diarrhea, you see the cysts. If you look at them on a microscope, which we have the slides, and then you see the cyst. Another person comes and pick up, I poop in a body of water in a river. Somebody down the river grabs the water and drinks the water from the river, for example, does not filter it, and then the cyst goes into the intestine. They go through excisment. You remember those terms we talked about it earlier? So they go through excisment, they multiply by mitosis, binary fission, right? Multiply and multiply and multiply. Then I develop diarrhea, and then I start releasing the cyst. While they're dividing, for example, this is the Giardia, the flagellated Giardia trophozoite stage, divides, some of them become a cyst. Some of them become another trophozoite. Remember mitosis, one cell becomes two cells? So some of those cells become a cyst, and the cyst, they leave. Of course, in the feces that you're looking at it on the microscope, you can see some trophozoites. Somebody with diarrhea, you grab their feces and put it on a slide, you will see some trophozoite stage. This is a trophozoite stage right here. This is a trophozoite stage with 2 nuclei. This is a cyst stage with 4 nuclei. Okay? The cyst stage has 4 nuclei, and the trophozoite stage has 2 nuclei. And then, of course, the cyst stage does not have any flagella, right? You know, but flagella are here, so on and so forth. Okay, phylum Parabasalia. These are the Trichomonas vaginalis, the 2 species that we do have slide, and you should know about them. One of them is Trichomonas vaginalis. This is the only sexually transmitted protista that you should worry about for this class. Okay, trichomoniasis is the name of the disease, and it is transmitted by sexual transmission. Sexually, it's transmitted from male to female. Okay, usually male do not develop much of a problem, but females, they have a foul smell, vaginal discharge, and it is more severe in female than male. In male, again, the numbers have to be high. Something about giardia, I'm sorry, let's go back. Talk about giardia. In every disease, guys, let's have a heart-to-heart talk. And let's say, I mean, you never said this. I did say it. But it's not on my PowerPoints, but I did say it. Okay. For all diseases, all pathogens, if they're studying them, some of them is under intensive research, you have to ingest or you have to get that pathogen a set number that causes disease. Am I making some sense? Let me give you the number for giardia. For giardia, if you get 100 of them, then you develop diarrhea. If you get 50 giardia, you get 60, you get 70 giardia cyst, you will not develop diarrhea. So the magic number for giardia is 100. About 100 cysts you must ingest in order that you develop the disease diarrhea. Am I making some sense? And that is true for all diseases. All the way from common cold, you catch a cold not by one rhinovirus. You must ingest a few of them, inhale, ingest, whatever it is, then you develop cold. And that is true for all of diseases. So when I said, in case of Trichomonas vaginalis, the numbers usually for female is lower, okay, than the male. Maybe because the distance of the urethra is longer. Okay, so that's maybe the reason. I don't know. But anyhow, females get vaginitis, they get trichomoniasis, and so on and so forth. The other one is trichonympha. We do have a slide of that. That's a termite gut. It's a mutualistic type relationship with termite. Termite eat wood, but if you remove these guys from gut of termites, they starve to death. So these guys in the gut of termites, trichonympha, they break down cellulose to single sugar molecules, so the termite can use it and also themselves can use it. That is the mutualistic relationship they have with the termite. And again, okay, we do not have this one, do not worry about it, but this is in the lab we have trichonympha. Large Golgi bodies, Trichomonas vaginalis, already gave you that, is that right? And then what else? Giant amoeba, we do not have it in the lab, but that's another organisms that they remove them from amoeba phylum and they put them in here mainly because of the DNA similarities with these guys. Trichonympha, we do have slide of that one. We do not have slide of giant amoeba. And flagellate protozoan that cause African sleeping sickness. So that would be a nice question. What are the flagellated organisms? Amir is afraid of, do you remember you had a question? Whatever, Sarah, Adam, whatever his name was, is afraid of amoeba. Now you might get a question. Amir is afraid of flagellated organisms. What are flagellated organisms that cause disease? So that would be, here, this one, I just have a few of them. This is Trypanosoma, which belongs to what phylum? Quiz question. Euglenozoa. And Trichomonas vaginalis belongs to what phylum? Parabaceae. Okay, so for your lab practical exam, I would like to ask the classification questions. You should know. Okay, major protozoan taxa. Other species, phylum Dinoflagellata, other species of Dinoflagellata are harmful to and toxic to many organisms. The P silent, Pfisteria, Piscicilia, again has both amoeboid and flagellate forms among FLA. I think I've mentioned that. 20 body types generally feed on bacteria and algae where they can temporarily use energy stores in chloroplast. Releases short-lived toxins when exposed. Okay, exposed to fishes. Extract that can be stun, kill fish, create lesions, affect fish along the Atlantic coast of the United States. Here is go red tide, which you mentioned this morning. And remember, I want you to know it belongs to phylum, Dinoflagellata. And I gave you 100 species. We do have in the lab a serratium. Serratium, I should have moved it and put the serratium right here. It belongs to this phylum, Dinoflagellata. Okay, here are, again, here is the serratium, and here are some of the species which we do not have in the lab of Dinoflagellata. Epicomplex. Today we'll finish everything and we'll leave Toxoplasma gondii for Thursday, and a week from today you have your embryology and Protista exam. And on Thursday it is your presentation. Is that right? The first one. I hope you'll do a good job. It's the first one. You don't know. I might not be too lenient because I already said everything that's supposed to be said. Why do I have a picture of a tank in here? The reason for that, because the way these guys move is they call it by gliding. So you have cell membrane here. Underneath of cell membrane, there are these cytoskeleton, these protein molecules from Bio-1. Cytoskeleton are made up of protein molecules. Is that right? And the way they're proposing that the cell membrane glides over these protein cytoskeletons. These protein molecules, and that's what they call it. These organisms are moving by gliding, just like the wheels of a tank. One semester, I know everybody have seen a tank. I asked a student, have you seen a tank? He said, no, I have not seen a tank. I said, have a picture of it? He said, no, I have not seen a picture. I said, wait a minute, you're very innocent. You're just like my son. My son is 4.5, 5 years old. I said, you're a very innocent person. So if you have not seen a tank, you don't know what tank wheels, you all know what tank wheels look like. Okay, so there's a belt goes around these wheels, and that's how epicomplexia move, they say. All of the members of this phylum are parasitic. You have not seen that yet, right? Am I making some sense? They have a specialized organelle called apical complex. You remember at the beginning of last time, I said Protista, some of them have specialized organelles. The specialized organelles that I'm mentioning right now, it is called apical complex. There are a few structures inside of that organelle that I'm only mentioning two of them, rope trees and micronemes. There are a few more. There are a few more, but those two I'm hoping you'll be familiar with. And these are the ones, the apical complex, the theory has it that you have the host cell right here, and this is the apical complex, and they digest, they release molecules, enzymes, what they have, and they digest away the cell membrane of the host so they can get access inside of the cell host, the host cell. Am I making some sense? So they gain access inside of the host cell. And that's what the function of apical complex is, they say. Okay, so now you know what the function of apical complex is. It's to gain access to host cell. Pseudopod, you will see here in a minute that some stages of this organism have pseudopod. Some stages of the life cycle. Some stages have pseudopod. And in some intracellular stages. Flagella in gametes of some species, the gamete stage has flagella. Okay, and you will see that sexual and asexual reproduction. Typical of Protista. Some require intermediate host. Some developed oocysts or spores, and I'll go over that. You remember Giardia, we just got done talking about Giardia or Euglena. Euglena is free living, Giardia is parasitic. Am I making some sense? They both have cysts, right? However, these guys, Epicomplexa, have oocyst. Oocyst. That's the way you pronounce it, oocyst. I say it twice. They want to make sure, you know, they do not want to call it oocyst. They call it oocyst. So anyhow, so they develop oocysts. Here is the apical complex that I was talking about. You have polar ring, conoid, subpellicular microtubules, micronemes, rope trees, and micropores. So these are some of the structures of the apical complex, but I'm asking you only to know two of them, right? So again, they have Golgi apparatus, mitochondria, nucleus, just like regular normal cells. And then this is a oocyst structure, and inside of it you could have 4 or 5 sporocysts. Not 5, I'm sorry, 4, 2, 4, 8. Okay, those are the structure. Class Coccidia, again, for this exam, I do not want you, do not worry about class and subphylum or anything else. Just worry about the name of the phylum and genus species, right? Because it's such a lot of information. But this is one class that a lot of people, if you go out there, you talk to any farmer or you talk to a doctor, they might say this is a coccidiosis. This disease that you have is a coccidiosis, and you're wondering what the heck is coccidiosis. Here it is. Let's run it by. Okay, so that is a common term that you will hear as far as epicomplexa goes. Coccidiosis. Okay, coccidia. Pyroplasm in cattle water, Babesia. Okay, Babesia bigemina, or Babesia bigemina is called Texas red water fever. What happened the first time they saw it, they noticed it in cattle in Texas. That's what they call it, Texas. It can happen in California. It can happen all over the United States, all over the world. What happened? The cattle urinated, and the urine was bloody, was red. So, and the culprit is Babesia bigemina. We humans can get Babesia bigemina, and then the similarity between this one and malaria is very close. People, doctors, usually misdiagnose this with malaria. A person who has Babesia is less severe, less dangerous than malaria. So people who have malaria are diagnosed, maybe they are misdiagnosed. They have Babesia. Okay, so anyhow, that's another. Those of you who did not mention coccidiosis, that was not you, right? No, it was not you. Coccidiosis, here is one of them, Babesia. Okay, the other one, cryptosporidium. I will talk about cryptosporidium a little bit later on, but cryptosporidium causes severe diarrhea. What I mean by severe diarrhea, in case of Giardia, you get rid of it, your immune system kicks in, and within 24, 48, 3 days, your diarrhea will stop. However, with cryptosporidium, that's not the case. You will have severe diarrhea for days, and then you have to receive medical attention in order to stop it. The IV, the anti-protozoan drugs, and so on and so forth. So the case with cryptosporidium is more severe. Those of you who had diarrhea questions this morning, so cryptosporidium was skipped. And people who online, I asked the same questions, the online people get it, they skip cryptosporidium. Muhammad, is this on? Can you make sure it is on, the red light is on?
Yeah, it's recording.
Thank you, sir. Okay, how did we get here? Yes, cryptosporidium. Coccidia, they are intracellular organisms. They go inside of the cell. Eimeria and Isospora cause coccidiosis. Okay, and then Isospora in AIDS patients is it can cause death. Of course, nowadays it's getting better now with the medications and drugs. They can control this organism. And then Eimeria tenala in young foals, if you talk to any chicken or poultry farmers, chicken or turkey farmers, they will let you know that this is the nightmare. I've heard stories, I don't know, I've never seen it. I've heard stories, my teachers were telling me, that when the farmers got up next morning, all of the chickens, millions, hundreds, thousands of chickens were dead next morning. That's how fast it can spread and how fast it can kill chicken. Okay, so that's why in the United States, I believe, because of that, they give chickens a lot of antibiotics. They give them a lot of antibiotics. And you know, chicken eat each other's poop. And that's how it's transmitted. So the chicks will release the oocyst in the feces. Another chick comes eat poop of the chicken with diarrhea, and then of course they all have diarrhea and they die within 24 hours. And that's why in the United States they give chickens a lot of antibiotic to prevent this. Okay, high dosage, different type of antibiotic. Okay, reproduction of Eimeria, let's go over it. Schizogony, it's a cycle, so we might as well start with schizogony, and you already know what schizogony is. I don't have to go over it, that's the quiz question today. Okay, so schizogony forms the gametes, microgamete and macrogamete. Microgamete and macrogamete come together, fertilization, right? Microgamete, macrogamete come together, and then after fertilization, in the poop, you have oocyst. Am I making some sense? And schizogony is happening in the intestine, if you did that. Okay? Then in the intestine and feces, what happens? Then another chick comes eat the oocyst in the feces, as I said. And what happens in the oocyst? Sorry about that. In oocyst, you have sporogony going, right? I hope I'm not confusing you. In the oocyst, one nucleus becomes 2, and 2 becomes 4. It depends on the species inside of the oocyst, right? And then what happens, another chick comes to eat their sporocyst, and in the oocyst, and 8 sporozoites outside of the host. Where is it, I guess? New host ingests a sporulated oocyst. The new host ingests sporulated cellulosis. The oocyst goes to the intestine, and in the intestine, they find those 8 ones separates. They get the excisement occurs. I hope I'm making some sense. If not, during lab, let me know. I'll go over it. I'll draw it on the board. And then they get out, 8 of them. They get out in the intestine of the host. Each one of them undergo binary fission. They multiply, 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 of course, in the schizogony stage. The schizogony, they find a host cell, not on the binary fission, sorry about that. Again, I'm not focusing on what I'm saying. Right here, they gain one of those 8. There are 8 of them. 1, 2, 3, 4, 5, 6, 7, 8. Those 8 separate from the oocyst. They gain access inside of the host cell, and then schizogony is what? Multiplication of the nuclei without cytokinesis. So when then the cell is almost dismantled, destroyed, then they form the cell membrane. I'm drawing schizogony for you. Cytokinesis occurs, and of course the host cell dies. The host cell dies, they get out, and they go infect another cell. I hope the life cycle, I made a few mistakes talking about it, but I hope I'm straight. Okay, the next one, Toxoplasma gondii in AIDS patient, it could be a problem. Based on statistics, half of us in this room have it. 50% of population have Toxoplasma gondii, and you don't even know. We'll go over that. 50% of population, half of us in this room, half of Delta College, this is a small sample. I cannot make a claim like that. But half of Delta College, City of Stockton, this is a good size. Large, those of you at statistics know what I'm talking about. The larger the sample, the better your finding is. Okay, so half of people in California, half of people in Stockton have Toxoplasma gondii, and we don't even know. Okay, and definitive host is cat. And what does definitive host mean? A definitive host means the sexual reproduction occurs, where the male microgamete and macrogamete come together. That's called definitive host. Okay, and you will see that in case of Eimeria, definitive host and intermediate host is same, chicken. In case of Isoscora, Eimeria, same host. But in this case, wild cat, domestic cat is the definitive host. Possible intermediate hosts are, yes, possible, I didn't give it to you. Human is one of them. Okay, goat, sheep, these are all possible. Chicken, pigs, those of you who eat pork. Okay, so those are all possible intermediate hosts. Plasmodium, malaria. Malaria transmitted by mosquito. And guess who is it? Speaking of definitive host, guess who is the definitive host in case of malaria? Mosquito. Mosquito, where the sexual reproduction occurs. Microgamete and macrogamete get together inside of the gut of mosquito. Why is it mosquito? I mean, I don't know. Ask the big one. I'm innocent. Is that right? Have you heard that joke? I'm innocent. Don't worry about it. There are so many jokes I gotta say, and I gotta say it to you guys, but not on the video. Is that right, Aisha? On the video it will not be fun. A lot of people watching it worldwide, and then I'll get into trouble. Okay. Sporozoites injected to human by insects. Of course, the insects we're talking about, mosquito. Sporozoites in liver initiate schizogeny. I've got to show you the picture. The most dangerous, there are 4 different Plasmodium species. The most dangerous one to human is Plasmodium falciparum. If somebody's infected, and most I've heard on the radio, again, I don't keep up with these things anymore, but I've heard on the radio when I was driving to work one day that the most common one, unfortunately, is Plasmodium falciparum. That's very unfortunate, but anyhow. That's based on what I heard. But again, penetrate red blood cells after 1 generation in liver. In red blood cells, trophozoites feeds on hemoglobin. And then sexual reproduction, microgametes and macrogametes. Mosquito ingest the gametes. And fertilization of cyst. I'm waiting for the life. Here we go. You should know this. Oh, you're going straight for it. It's a cycle. It doesn't matter where I start. Might as well. Let's start with human. Or if you want me, I can start with mosquito. United States had problem with this, with malaria. But they were, you know, this land that we're living in it, so I think we should be very thankful to the people very early on. They worked very hard in here to get rid of malaria. Back in 1930s, they had malaria, and they went and they sprayed all of the swamps, rivers, lakes, everywhere that this mosquito was. Now they're trying to implement that to the rest of the world, the countries who have problem with it, malaria. But the politicians are so corrupt. They get the money from the United States and from Western countries, but they put that money in their pocket. And of course, spraying gets in the back burner. So that's why we cannot fight malaria. That's one of the reasons we cannot eradicate malaria from the world, because I think, again, my personal opinion, corrupted politicians. That's what I think. They get the money from U.S. and World Health Organization, World Bank. They get the money to spray. So we had that in the United States. Malaria did exist in the United States, but they got rid of it. They worked on it. Okay, so having said that, let's go over this. What happens when these guys, they call them ingested sporozoites, the sporozoites come inside of human? The first place they go, you learn this in first chapter material, the first place they go into is the liver. In the liver, the hepatocytes have the right receptors for them, so they gain access, they go inside of the hepatocytes and undergo schizogeny. Sporozoites in there and then undergo schizogeny. And I did draw it for you, schizogeny, already in there. So they destroy liver cells, and they get outside of liver cells, right here, and they call them merozoites. And merozoites then, after one generation, in case of Plasmodium falciparum, after one generation in liver cells, they go find red blood cells. That's the slide you have in the lab. You have slide of red blood cells in the lab with Plasmodium inside of them. Okay? So, inside of red blood cells, they do the same thing and destroy the red blood cells. When the red blood cells are destroyed, then the person have chills, fever. That's one of the signs and symptoms of malaria. People have chills and fever. Headache. Those are all because red blood cells are being destroyed. And they come back. Of course, by the time they go find another set of red blood cells, the chills and fever goes away. But this happens so often, so the person will die eventually. Okay, so chills and fevers go away, but comes back again. Intervals. Again, I don't want to bore you with a lot of details. Okay, then what would happen, a mosquito comes and take a blood meal from this infected person, and then there are microgametes and macrogametes, some of these that are going into the red blood cells and multiplying. Some of these become microgamete and macrogamete, and the mosquito comes, take a blood meal, which has the microgamete and macrogamete, and inside of the mosquito ingested gametocytes, microgamete and macrogametes, come together, the female gametes and the male gametes become amoeboid, like an amoeba. Am I making some sense? And then they are fused together, fertilization. Do not worry about that term, but I'm referring it to as an oocyst. You do have a 2x2 slides. 2x2 slides as I'll show it to you today. Well, not today. It's coming up here in a minute. So then the oocyst, inside of it you have sporogeny. We talked about that. And when inside of the gut of the termite, when the sporogeny ruptures open, then you have the sporozoites, and sporozoites, when the mosquito goes take a blood meal from another person, and a not infected person, a healthy host, then it transmits those sporozoites into the healthy person, and the cycle goes on and on and on from one person to the next person to the next person. Of course, the problem with this in Africa and other countries, that some of the wildlife, some of the domestic animals act as a reservoir. Okay, they act as a reservoir for this parasite, so the mosquito can jump from a cow or a buffalo to a human and then transmit it. But even in big cities, this is a problem. That could be a rural area, but big cities is a problem because the mosquito transmit this from human to human. Glenn, I'm ready.
Are we supposed to know whether it's indirect or direct life cycle?
This is indirect life cycle. The one thing that I have not mentioned so far, thank you so much, Glenn, when you have a mosquito or another organism involved in the life cycle, this is indirect life cycle. In case of Giardia, it's a direct life cycle. It gets out from me, and another human have to pick up from the feces. It's a direct life cycle. There is no mosquito involved. In case of Cryptosporidium, direct life cycle, feces to another host. Giardia, what else? All of these coccidia that I talked about so far, except malaria and toxoplasma, no, you need cat. This, you need mosquito. Trypanosoma, you remember trypanosoma? Tsetse fly, leishmania, those are in direct life cycle. They are indirect life cycle. They are not direct life cycle. Direct life cycle, it means from host to host in the feces, Trichomonas vaginalis, direct life cycle, right? From human to human. No mosquito involved, nothing else is involved. Anyhow, whatever comes up, then we can discuss. Cryptosporidium parvum, I gave you the name of species as well. Genus and species. Before last year or the year before, I just said Cryptosporidium, but it's Cryptosporidium parvum. I have seen with two U's, P-A-R-V-U-U-M. I don't know, maybe those scientific papers I've seen, they misspelled it, or maybe there are two spellings. But anyhow, live in small intestine.